let's pinpoint some of the benefits of cranberry beyond uterine tract infections.
The proanthocyanidins, or PACs, actually, the majority of those compounds never leave the lumen, or the intestinal tract.
Really?
Okay.
That's fascinating.
Yeah, so most folks think, well, let's, classic absorption. If we consume it, it goes into the stomach, it crosses the barrier, goes into the blood system, makes its way through the whole body. Proanthocyanidins, or PACs, have a very specific way of functioning in that they don't cross that barrier. So they stay within the lumen of the stomach. And so I would say probably 90 to 95% of the total PACs never leave the lumen.
That's incredible, because when you think of Crohn's disease and when you think of irritable bowel syndrome, just having that remain in the gut would seem huge. So when do you think something may be coming out that will be more scientifically proving that that can help and people can take a supplement?
Well, one of the interesting studies that came out, and it was maybe 2 years ago, I think it was 2012 or 2013, there was a study conducted at the University of Wisconsin at Madison, and the study actually looked at the effects of proanthocyanidins in an intestinal environment. And what the researchers found is that the proanthocyanidins actually increase barrier function of the intestine. So what does that mean?
What does that mean?
Exactly. What we're saying is that, okay, the intestine is a barrier. There's certain things cross and certain things don't cross.
Yeah.
And one of the things that builds up on the intestine is mucus. So we think about the mucosal surfaces like the mouth and the respiratory system and the intestine. And we found through studies like this that PACs actually increase the amount of mucin production in subjects that are fed simplified diets. So it's showing us that there is some actual mechanisms by which these PACs interact at a gut level.
Does it almost form like a force field?
Well, it seems like that.
Yeah.
So when we think about the intestine, what happens with the mucosal layer is that it actually houses other immunoglobulins and antibacterial compounds. And so if you think about having a big thick layer, you think about having more protection or more ability to combat bad bacteria that may make their way into the digestive system.
That's amazing. Now, there are two classes of PACs found in cranberry. How can you explain that for us?
There are two classes of PACs, and we refer to them as either soluble or extractable PACs, or insoluble or non-extractable PACs. And when we talk about extraction, a juicing process is a really good way to think about this. If we take a cranberry berry itself, a fresh berry, and squeeze it under a lot of pressure, get the juice free. What comes with the juice are soluble PACs. They were able to be removed in a liquid form, in a liquid medium, from the fruit itself. Now, there's another class that remains with the fruit fiber and proteins, the residual that we call press cake or pumice. Those are insoluble PACs. Doesn't mean they're different in structure. What it means is they're associated with other parts of the cranberry, such as the fibers, so they'll be carried around. So really what you're doing is you're splitting one big group, proanthocyanidins, into two smaller groups.
Let's talk about gut health, because I know that's a very important aspect of cranberry.
So gut health is very interesting. One of the projects that we've worked on recently is looking at the ability of cranberry PACs to inhibit the invasion of epithelial cells. These are the cells that line the intestine. And what we're looking at is, can we prevent E. coli, or pathogenic bacteria, from actually invading the epithelial cells and setting up a colony there and propagating?
Before we go on, PACs.
Proanthocyanidins.
Okay, just so everyone
Yes,
knows, but they're also
absolutely.
called PACs.
They're also called PACs, and they have a unique structural feature, an A-type linkage, that defines cranberry proanthocyanidins. So it sets them apart from other proanthocyanidins.
Are cranberry PACs different from other fruit PACs?
Yes. The biggest difference really is cranberry PACs have A-type linkages or A-type bonds. Other packs like grapeseed extracts or apple packs tend to have primarily only B-type linkages. And again, this is how, think about a chain. How do we connect the links in a chain? A-type links or B-type links. That differentiates the packs.
So what benefits then will people see as far as gut health and cranberry?
Well, what we are learning is that the specific A-type structure of the packs from cranberries interacts with surface factors on pathogenic bacteria. So what it's doing is it's almost docking, right? Is that there's a specific site on the bacteria and there's a specific site on the pack. And these two guys come together and they fit. And what that fit does is prevent the E. coli or pathogenic bacteria from actually gaining access to the cells of the body.
Thank you so much for joining us today. Fascinating stuff.
Thanks, Jennifer.